Click the bell icon and subscribe to our channel so that you will not miss any classes.
Hello, welcome to class. We shall discuss in this class the direct methods of gene transfer or vectorless gene transfer methods. Vectorless gene transfer methods. Generally, we studied under the discussion of preparation of competent host. Recombinant DNA is made to enter into the host. Okay, so where now the recombinant DNA is only used, not the direct gene. There are some methods available in biotechnological processes where we can directly isolate the gene, and isolated gene can be introduced directly into the genome of a host. So for that various methods are available, and don't think these methods can only transfer the gene. It cannot be used for transferring only the gene. These are also used for transferring the recombinant DNA also, that is DNA with a vector. Gene with vector can be transferred. Gene without vector can also be transferred. So that is the case. Okay. So here in these methods, it is possible to transfer of recombinant DNA as well as gene or DNA without vector can be transferred successfully into the host. Okay, so various methods have been available. So first one is electroporation. Electroporation. So as the name suggests, there will be creation of pores under electric field. So what they will do is here, first the host is taken, host of our interest. We have studied from the previous that animal, plant, bacterial or fungal cells, anything. So host is along with the recombinant DNA or directly the gene. Will be made into suspension. Usually the suspensions are made up of saline. And to this suspension, electric field of very low voltage is given for short period of time, very short period of time. So when the electric field is given to this, in the host there is a creation of force. So through this force the DNA can enter into the host genome. So this is the method of electroporation. This is one method. Then the second method available is microinjection. This microinjection technique is popularly used for transferring the genes into animal cells. Okay, so here microinjection, what they will do is they will take a host cell, preferably the ovum is taken. Okay, ovum will be isolated for conducting this, or any other animal cells can also be used. Popularly used, I am telling now. So here the animal cells will have the extrinsic nucleus. So this cell will be kept under high magnification power microscopes. So they are working with a single cell, right? So the procedure of microinjection is conducted under high magnification microscopes. So this procedure is very expensive because the instrument itself is very expensive. So commonly it is not being used. So under research works which is very necessary, there they make use of microinjection technique. Okay, so this cell will be kept under microscope of high magnification power, and this cell will be held with the section pump or section holder. So here the section is created, that is the pressure is created. So this is holding instrument. Okay, so this is for holding. So this will hold. There is a creation of small amount of section pressure here. So the section pressure should be moderate. If it increases, then it will suck the cell into the instrument. If it is very low, it will leave the cell. So that is the problem. So moderate section pressure will be created in this holding panel. Then micropipette is available of very sharp end like this. Okay, so at the end of this micropipette, they'll add the recombinant DNA or the gene. So directly gene can be used, right? So then by observing under the microscope, this DNA is injected into the nucleus. So we cannot inject into the cytoplasm, leave it as such. So as like we do for the prokaryotic cell, that is not the case here. Here this DNA should be injected directly into the nucleus of a cell. Okay, so this is the method of microinjection. So this is mainly for the transfer of genes into animal cells. Then the third method available is gene gun. Gene gun, or it is called by many alternative names. I'll write what are possible names now. Gene gun, or it is called particle gun, or it is also called shotgun, or it is also called biolistics. And this method is used for transferring the genes popularly to the plant cells. For transferring the genes to plant, they make use of gene gun method. So here the gene gun is an instrument which is having the chamber for the collection of the recombinant DNA or the gene of our interest. Okay, so here like this. So this is the holding panel where we can load the gene of our interest. So here the gene of our interest will be loaded. Gene of our interest are loaded not like that. That means only the gene of our interest are not loaded. Here the gene of our interest or the recombinant DNA will be coated to gene or recombinant DNA is coated to gold or tungsten particles. Gold or tungsten particles of minute size. Minute size means they have the diameter of 1 to 2 microns. Okay, their size. To that particles, so suppose if it is a gold or tungsten particles, this will be coated with the gene of our interest or the recombinant DNA, and this will be loaded here. Okay, so this will be loaded. Right, then This chamber, below this chamber, there is a holding panel for the host. Usually, as I told, it is this method used for transferring the genes to plant. So they place the leaf here. The leaf will be placed here and this chamber. Okay. So here, these gold or tungsten particles after loading will be bombarded with. So here section pressure will be there. So under that section pressure, high pressure or bombardation will be given. Okay, bombarded with high speed. High speed here is of approximately 1400 feet per second. So this, with this speed, the gold or tungsten particles which are coated with the DNA or gene will be bombarded. With that high speed, they will enter into the genome or the cell of the plant. So this is the method of gene gun or particle gun. So apart from this, some disarmed pathogens are also used for transferring the genes into the host. For example, we have studied already in the previous videos that the Agrobacterium tumefacium, that is the TI plasmid, is disarmed. Okay, and it is used for transferring the genes to dicot plants. And also retroviruses. Retrovirus have the capacity to cause the cancer. So that cancer causing capacity is remote. That means it will become disarmed then. So it will not harm the host. And that can be used for transferring the genes to any of the host. So that is the fourth one. Just have a count. Some disarmed pathogens like Agrobacterium, retroviruses, SV40 virus. So these are the pathogenic viruses or pathogens, we can say. They are made non-pathogenic or they made disarmed, and then they can be used for transferring the genes. So this is the fourth method of gene transfer, vectorless gene transfer methods. Okay, so here is the count of all the types of vectorless gene transfer methods available. And in the next class, let us discuss about the next tool in recombinant DNA technology, that is PCR.